In this video, I'm going to draw out the general steps for the glycolysis pathway. There won't be any structures in this video, so if you need those, there's another video on glycolysis with more detail. Glycolysis is an energy conversion pathway, and the primary goal is to break down 1 glucose into 2 pyruvates, generating a yield of 4 ATP and 2 NADH. The pathway is exergonic, releasing energy to drive ATP synthesis. It is important for essentially all of the cells of the body, but it is also used in all forms of life, from prokaryotes to bacteria and animals. Different cells rely on it to different extents. Our brain uses it for its primary fuel source under non-starvation conditions. Our muscles use it because it's a faster energy source than oxidative phosphorylation, which is important when we need immediate muscle contractions. Our red blood cells have no mitochondria, so glycolysis is the only way that they can get energy. There's no oxygen requirement. It has 10 steps and 2 phases. Phase 1 is prep and cleave, where we have 2 ATP used to set the molecule up to be split, while phase 2 is our payoff phase, where we get our energy in the form of 4 ATP and our pyruvate. Since we use 2 ATP in phase 1 and generate 4 ATP in phase 2, we have a net of 2 ATP per glucose. This pathway takes place entirely in the cytosol, so our first obstacle is getting the glucose into the cell. Glucose can't diffuse freely into the cell. To get it in, we'll use special transporters called GLUTs, standing for glucose transporters. Different tissues have different forms of glucose transporters, but I'm going to keep it to the general glucose transporter here. So these glucose transporters are bidirectional, and they go along a concentration gradient. As long as the glucose concentration in the cell is lower than outside, we can get the glucose in. Our glucose is in the cell. Now we have to trap it here. To do that, we're going to add a negative phosphate group to the 6th carbon of the glucose. This will give it a negative charge, and it will no longer be able to leave through the transporter. It will also increase its energy. Since phosphate groups are high energy, this will make things more reactive with the goal of getting us ready to split later on. This new molecule is called glucose 6-phosphate. How it gets the phosphate is ATP will donate the phosphate, becoming ADP in the process. And this hydrogen will come from being replaced on the 6th carbon by the phosphate. This is catalyzed by one of 2 enzymes, and both enzymes are kinases because kinases add phosphates. Hexokinase is in all cell, while glucokinase is only in the liver and the pancreas. For this reaction to happen, we'll also need magnesium as a cofactor. And the magnesium is just going to help things along by stabilizing the ATP during the phosphate transfer. Magnesium is going to be required by a lot of the reactions here, and all of the ones involving kinases. This reaction will only go one way. The carbons and the phosphate are staying in the same position, but things are getting moved around internally here, and our product is going to be fructose 6-phosphate. This reaction will be reversible, and the direction will depend on the concentration of glucose 6-phosphate and fructose 6-phosphate. For our enzyme, isomerases are enzymes that move things around within a molecule. They don't add or remove anything, and it will still have the same molecular formula. What is happening here is we're getting our molecule ready to be split, because the fructose is better able to be symmetrically cleaved as a pentose than glucose is as a hexose. The name for the enzyme that catalyzes this step is phosphoglucose isomerase. In our next step, the phosphate is staying on our 6th carbon, but we're also getting one on carbon 1. ATP again will donate it, becoming ADP. Our magnesium will stabilize things, and this reaction is only one way. The name for this enzyme is phosphofructokinase 1, or PFK1. From the name we can tell what it does. Kinases add phosphate, and we're adding it to a fructose. For this new molecule, we're going from a fructose 6-phosphate, adding another phosphate to carbon 1, we get fructose 1,6-bisphosphate. This phosphate means the phosphates are on 2 separate atoms, in this case carbon 1 and carbon 6, while with diphosphates like ADP, we have the phosphates together in a chain. This enzyme, the PFK1, catalyzes our rate-limiting step. It is our checkpoint for glycolysis to make sure that the cell actually needs to continue, and the reaction only goes one way. So we've gone from a glucose, added a phosphate to it, changed it to a fructose, and added a second phosphate. These two added phosphates have increased this molecule's energy, and it's now ready to be symmetrically cleaved between carbons 3 and carbons 4 to give us two 3-carbon molecules. This is done by aldolase in a reversible reaction. It produces dihydroxyacetone phosphate, or DHAP, and glyceraldehyde 3-phosphate, or GA3P. DHAP has the phosphate on carbon 1, while GA3P has the phosphate on carbon 3. These are isomers of each other, and since they're isomers, they can interconvert with the enzyme triose phosphate isomerase in a reversible reaction. Balance between the DHAP and the glyceraldehyde 3-phosphate is much higher towards the DHAP, with it making up around 96% of the products, as it's more stable than the glyceraldehyde 3-phosphate. However, only the glyceraldehyde 3-phosphate will move down this pathway, but as it moves down, it shifts the balance between the two and more DHAP will convert to GA3P. This next reaction will be reversible, and it will be catalyzed by glyceraldehyde 3-phosphate dehydrogenase. Dehydrogenases catalyze redox reactions. Another phosphate will be used, this time in the form of inorganic phosphate, also called orthophosphate. So with that, because it's a redox reaction, we know we're going to need something to accept our electrons, and that's going to be NAD+. This NAD is going to be required, or there's going to be no reaction, and it will have to be regenerated later. The GA3P is oxidized, NAD accepts the electrons, and it's reduced to NADH. We will also release a hydrogen. Our product will have a phosphate on the carbon 3 and a new one on carbon 1. This gives us 1,3-bisphosphoglycerate, or 1,3-BPG. This is a high-energy intermediate, getting us ready to make ATP. In our product, we have that hydrogen on its own. An organic phosphate exists as HPO4 2-. So when this reaction takes place, the hydrogen is replaced when the phosphate attaches. Our high-energy intermediate, 1,3-BPG, now has a bond that's very strained, making it reactive. The enzyme phosphoglycerate kinase, along with its support magnesium, will catalyze our first ATP. ADP will come in, accept the phosphate to become ATP, and this hydrogen will go where the phosphate was. This reaction goes both ways. If you're wondering why the enzyme is named for the reverse reaction, it is also used in the gluconeogenesis pathway going in the reverse direction. Now, which carbon did that phosphate come from? Carbon 1. So that gives us 3-phosphoglycerate. This step is substrate level phosphorylation, because our substrate, 1,3-BPG, is what donated the phosphate. For our next reaction, the phosphate is moving to carbon 2 position. We're getting a repositioning of things, which is reversible. Mutases are a class of isomerase that reposition groups within the molecule. This is done with the enzyme phosphoglycerate mutase, or PG mutase, with its support magnesium to stabilize the reaction. So we're going from 3-phosphoglycerate with the phosphate on the 3-carbon, and we're moving that phosphate onto the 2-carbon, so that gives us our product, 2-phosphoglycerate. The phosphate is staying put for this next reaction, but we're going to remove a water molecule. Catalyzed by enolase, we're going to get the product, phosphoenol pyruvate, or PEP. This is another molecule with high transfer potential, so we're getting ready to get rid of that phosphate. The support magnesium will stabilize an intermediate of this reaction, and this reaction can go both ways. And here we are. our last ATP being produced. ADP will accept the phosphate to become ATP. Hydrogen will take up that empty spot, and our final product of pyruvate is made. This is catalyzed by pyruvate kinase, and it will require the support magnesium as well as potassium this time, which will help stabilize and orient things in the enzyme. This reaction only goes one way. With the PG kinase, I had said it was named in reverse because another pathway uses it in the reverse direction. But pyruvate kinase only goes one way. Reactions that happen one way in the body won't necessarily happen one way in the lab. Reactions will go in the direction that is energetically favorable to maintain that reaction's equilibrium. So reactions that have a preference for one way under physiological settings may have a preference for the reverse under lab settings where they're discovered. So enzyme names will give an indication of what is happening, but you won't always be able to tell which way the reaction goes. So this is an overview of the entire pathway. I had said we would generate 4 ATP, but we only saw 2 used in the prep phase and 2 created in the payoff phase. Everything Ga3P down will happen twice, because we had that 6 carbon split into 2 3-carbon molecules, and each half will go down. We haven't yet got our NAD back, and this can be regenerated under different ways, depending on cell conditions and organism. Under aerobic conditions, we get our NAD back when NADH donates its electrons to the electron transport chain. For our pyruvate, it can have multiple fates depending on the oxygen level and organism. If we have aerobic conditions, the best case for our ATP production is going to acetyl-CoA and the citric acid cycle by losing a carbon in the form of CO2. The enzyme for this is the pyruvate dehydrogenase complex, but we won't get our NAD back for a while with this one. If oxygen becomes limiting, like in our muscles under strenuous exercise and some bacteria, pyruvate can go on to form lactate. Here we'll get our NAD back right away, and the enzyme that catalyzes this is lactate dehydrogenase. Some of the bacteria using this process are the ones that we use to make yogurt. In yeasts and some microbes, it will go through alcohol fermentation. The pyruvate will form acetaldehyde by losing a CO2 with the enzyme pyruvate decarboxylase. This enzyme will need TPP as a cofactor, which is a vitamin B derivative. In the next part of this reaction, we'll get our NAD back and produce ethanol with the enzyme alcohol dehydrogenase. This is the process that we exploit to get beer and wine. So that is our full pathway from glucose to pyruvate. Now it's on to its regulation. To regulate this pathway, our potential sites to regulate will be enzymes that catalyze irreversible reactions. Here we can see that we have 3 candidates in hexokinase, PFK1, or pyruvate kinase. There are 2 main ways that we have in the regulation of an enzyme. One is through covalent modification, where we add functional groups like our phosphates onto the enzyme to either inhibit or activate it. Another is allosterically, where something will bind to a site other than where the substrate binds to, which changes the affinity of the enzyme for the substrate, and it won't fit well in the active site. In order to know what will either activate or inactivate our enzymes, it's important to know what the goal of the pathway is. For glycolysis, it's the production of energy. Different tissues will have different patterns of regulation depending on their roles. Our muscles will depend on that energy for contraction, so they're mainly going to be regulated by the energy state of the cell, or how much ATP versus how much AMP we have. While for the liver, its main role is maintaining blood glucose levels. I'm going to start with PFK1 because it is our rate-limiting enzyme. This is our critical checkpoint for glycolysis, where the decision is made if it's actually worth going through the entirety of glycolysis, or if the intermediates can be shunted off to different pathways. So what's going to activate PFK1 is AMP, because it's going to indicate low energy levels. PFK1 has a site other than the active site where both ATP and AMP can bind to. If ATP binds to this site, it will inhibit the enzyme. But if AMP is able to bind here first, it won't inhibit the enzyme, but it will also prevent ATP from binding to it. It's also going to be activated by fructose 2,6-bisphosphate, but only in the liver. This comes from a buildup of fructose 6-phosphate being converted by an enzyme called phosphofructokinase 2, or PFK2, which is regulated by insulin and glucagon. This will keep glycolysis active in the liver after a meal. PFK1 is going to be inactivated by ATP because it indicates a high energy level, by pH in the muscle cells, and citrate in the liver. Hexokinase is the enzyme that catalyzes the addition of the phosphate onto the glucose to give us glucose 6-phosphate, and it's going to be inhibited by its product, glucose 6-phosphate. The reason for this is because if we have an increase in glucose 6-phosphate, it means that things aren't moving downstream, and it could indicate that PFK1 is being inhibited. PFK1 being inhibited would lead to an increase in fructose 6-phosphate, and because the next reaction up is a reversible reaction, we get an increase in glucose 6-phosphate. Because this next reaction up is with the hexokinase and it's irreversible, the high glucose 6-phosphate levels will inhibit the hexokinase so that the glucose in the cell will increase. And because the glucose transporters are bidirectional and work on a concentration gradient, if cellular glucose levels are too high, they won't bring in extra glucose, causing blood glucose levels to rise. Glucokinase in the liver is an isoform of hexokinase that has a higher Km than hexokinase does. This is important because the liver will move to store glucose when glucose levels are high. So we don't want to be storing glucose if there's cells that need glucose now. So glucokinase has a high Km, which means that it has a high threshold and requires high glucose concentrations before it will start working. This allows all of the cells like the brain, the muscles, the red blood cells, the things that really need glucose for their immediate energy needs, they will be the ones that get access to glucose before it starts being stored away. Pyruvate kinase catalyzes the last step in glycolysis, and it will be activated by fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate is the product of a rate-limiting step catalyzed by PFK1. So if we get through the pathway at that step to make fructose 1,6-bisphosphate, it's an indication that the cell needs the energy. So fructose 1,6-bisphosphate will activate the pyruvate kinase in a feed-forward fashion, kind of like opening a dam in springtime ahead of the winter melt. It's going to be inhibited by ATP, which indicates a high energy state of the cell, and it's also going to be inhibited by alanine, acetyl-CoA, and glucagon. For the inhibition by alanine, along with alpha-ketoglutarate, is capable of a transamination reaction to pyruvate and glutamate. So if we have enough alanine, we can easily have enough pyruvate. Pyruvate kinase is inhibited by acetyl-CoA because high amounts of acetyl-CoA indicate that there's a lot going through the citric acid cycle. And because we get a lot of energy from the citric acid cycle, we have an excess of acetyl-CoA. It's an indication if our energy needs aren't okay now, they soon will be. For inhibition by glucagon is our indication that glucose is gone. So if we don't have enough glucose, there's no point in keeping this pathway fully active. That is a lot of steps to remember, a lot of substrates and enzymes to remember in order. So if you panic on tests, mnemonics can help you through that. With mnemonics, we're just taking the first letter of each enzyme or substrate and making a story. For our substrates, we have a story about Grandpa Frank and Betty. Great-Grandpa Frank fights dinosaurs giggling. Betty prefers patiently playing piano. For our enzymes, it's all about famous people. High-profile people act too glamorous, prancing proudly every party. For the steps glyceraldehyde 3-phosphate down, I always found it helpful to count down. So to remember my substrates and the order they go in, I would go 1, 3, BPG is 1 plus 3 equals 4. My 3-phosphoglycerate is 3. My 2-phosphoglycerate is 2. My phosphoenolpyruvate is 1, because I like to remember that it still has a phosphate attached. And my pyruvate is none, because it doesn't have any phosphates and there's nothing left for the pathway. So I would count down from 4, 3, 2, 1, and none. And that is our full glycolysis pathway, where we've broken down our 6-carbon glucose into 2 3-carbon pyruvates, yielding 4 ATP from our payoff phase. So because our prep phase had used 2 ATP up, we net a total of 2 ATP. We also produce 2 NADH, which can go on to do different things.